Hey everyone, and welcome. Today, we are diving deep into the absolutely incredible world of proteins. And no, these aren't just something you think about in your diet, they are the literal, microscopic machines that build and run pretty much everything in our bodies. We're going to uncover how they're built, layer by fascinating layer, in a process that is just like biological origami. So that brings us to the big question, right? How in the world does a simple, one-dimensional string of chemicals manage to fold itself into these super complex, three-dimensional shapes that are needed to carry out life's most essential tasks? Well, that's the puzzle we're going to solve together today.
So here's how we're going to break it down. First, we'll get a handle on what proteins actually do. Then, we'll build one from the ground up, starting with its basic code, moving to its first folds, then assembling the final 3D model. And finally, we'll connect the dots on why that very specific shape is absolutely critical for its job.
All right, let's dive right in with our first section, life's microscopic machines. I want you to think of proteins as these tiny, highly specialized robots. Each one is built from a unique blueprint to perform a very specific function inside our cells.
And believe me, the jobs these proteins do are all over the map. They're the scaffolding for our bodies, like the collagen in our skin. They're enzymes that speed up chemical reactions. They're transporters, like hemoglobin carrying oxygen. They're messengers, like insulin. And there are bodyguards, antibodies fighting off invaders. All of these different jobs are only possible because each and every protein has its own unique, intricate structure.
Okay, so let's get to level 1 of building our protein, the primary amino acid code. This is ground zero. This is where it all begins. Think of it like a flat sheet of paper with the step-by-step instructions written out for our biological origami.
So what is the primary structure? Well, it's really just the straight-up linear sequence of its building blocks, which are called amino acids, all linked together in a long chain. Now, this sequence isn't random at all. It's spelled out perfectly by the genetic code in our DNA. This sequence is the absolute foundation for everything, and I mean everything that comes next.
So, how do you actually link these amino acids together? It happens through a really cool chemical reaction that forms what's called a peptide bond. Basically, two amino acids get cozy, a water molecule pops out, and boom, a super strong, stable bond is formed. You just repeat this process over and over and over, and you get a long chain called a polypeptide.
And you know, getting this sequence exactly right is a really big deal. Even one single mistake, just a tiny typo in this amino acid sequence, can cause the whole protein to misfold down the line, and that can lead to some really serious hereditary diseases. The instructions have to be perfect. Okay, so what happens next? We're on to level 2, the first folds and spirals. You see, that long straight chain of amino acids doesn't just stay like a floppy piece of spaghetti. It immediately starts to fold and organize itself into simple, repeating patterns. This is like making the first basic folds in our origami paper. This is what we call the secondary structure. It's all about the local folding of that polypeptide chain. And what's holding it together? Not those super strong peptide bonds, but much weaker hydrogen bonds. You can think of these like little pieces of tape, just temporarily holding those first folds of our paper right where they need to be. And you basically get two main patterns here, the α-Helix and the β-Pleated sheet. The α-Helix is kind of like coiling a ribbon into a spring, and it's held together by hydrogen bonds within that same chain. The β-Pleated sheet is more like making an accordion fold in a piece of paper, where the hydrogen bonds form between different strands lying next to each other. You'll find these helices in stuff like the keratin in your hair, and the sheets in the fibroin that makes up silk. Pretty cool, huh?
All right, we've got our basic spirals and our pleats. Now it's time to assemble the final masterpiece. This brings us to the next levels, where we go from a kind of folded chain to a fully functional, three-dimensional machine.
This next level is the tertiary structure. This is the final, overall 3D shape of a single polypeptide chain. All those helices and sheets we just talked about, plus all the other bits, they all fold up on each other to create this complex, compact, and most importantly, functional shape. At this point, our origami is a fully formed crane. It's ready to go.
So what's holding this incredibly intricate shape together? Well, it's a whole toolkit of chemical forces. We've still got our hydrogen bonds,
Sure.
but now we've also got super strong disulfide bonds that act like permanent staples. We've got ionic interactions that act like little magnets, and then there's this really important one, hydrophobic interactions. That's basically where all the parts of the protein that hate water hide together on the inside, away from the rest of the cell.
A perfect example of this is a protein called myoglobin.
It's a compact, ball-like protein that you'll find in your muscles. Its very specific tertiary structure creates this perfect little pocket that lets it grab onto and store an oxygen molecule until your muscle needs it. If it didn't have this exact 3D shape, it just couldn't do its job. Simple as that.
But you know what? Sometimes one finished origami crane just isn't enough for the job. And that brings us to our final and totally optional level, the quaternary structure. This is when multiple already folded polypeptide chains, each one its own little masterpiece, come together to form one giant functional machine.
And the classic example here is hemoglobin, the protein that carries oxygen in your blood. It's a perfect example of quaternary structure because it's actually made of 4 separate protein chains. All 4 of these parts have to assemble perfectly and work together as a team to efficiently pick up oxygen in your lungs and then deliver it all throughout your body.
Okay, let's bring this all home. In our final section, we're talking about why structure determines function. This is really the big takeaway, connecting that initial line of code to its real-world consequence.
At the end of the day, it all comes down to this simple but incredibly powerful principle. The sequence of amino acids dictates the final 3D structure. And it is that precise 3D structure that determines what the protein does, its function. So if you change the sequence, you change the structure. And if you change the structure, you change or you completely lose the function.
And this chart here gives you a great visual of the forces that are at play. You've got the super strong covalent bonds that create the permanent backbone and lock in key parts of that 3D shape. Then you have a whole bunch of weaker bonds and interactions that all work together to fine-tune and stabilize that final functional form.
So, just to recap the whole process, the primary level is defined by those strong covalent peptide bonds. The secondary level is all about those weaker hydrogen bonds making the first folds. And then the tertiary and quaternary levels use the whole toolkit, from strong disulfide bonds to all those weaker interactions, to get to their final, stable shapes.
And that right there is the story of protein folding. It's this beautiful, hierarchical process where a simple line of code gives rise to a complex 3D machine, with each level of structure building logically right on top of the one before it.
And let's not forget why all of this matters so much. This elegant process of biological origami is happening nonstop right now inside of you to create the enzymes, the structures, the transporters, and the defenders that are absolutely essential for you to be alive and healthy.
Which leaves us with a final and pretty fascinating thought. As we get better and better at understanding this process, could we one day become true masters of this biological origami? Could we actually learn to design and build our own custom proteins from scratch, maybe to cure diseases, or create new biofuels, or even build revolutionary new materials? The possibilities are just extraordinary. Thanks for joining me.